Okay. Good afternoon, everyone. Thank you for joining us today for the Research Against COVID-19 Call to Action webinar. My name is Jia Sun. I am the Associate Director of the Nanoscience Initiative and the ASRC Sensor CAT. So a few rules to start with. This session will be recorded. Please keep your microphone muted the entire time. The chat box is disabled currently, but it will be enabled during the Q&A session. So to submit your question, please type your name, organization, and your question. If we can't address all of the questions today, we will follow up with you offline. So a brief overview of the program today. First, we'll hear from the ASRC leadership. Then we'll hear 3-minute pitches from 5 different researchers within startups and academia. We'll continue to the Q&A session for about 20 minutes, and at 3 p.m. we'll end the program. So next, it is my pleasure to introduce Dr. Ryan Uihlein, the Director of the Nanoscience Initiative and the ASRC Sensor CAT.
Well, thank you very much, Gia. Am I muted? Am I good?
You're good.
I'm there. Excellent. Well, welcome everybody. We're very excited to see that so many of you could find time to join us for this call to action webinar today. And the purpose here is to try and see what we can do to address the COVID-19 crisis through science and see what we can do in our own small way to help with this. So what we hope to achieve today is to help connect and mobilize so a community of scientists and entrepreneurs with wide-ranging skills that can potentially be repurposed for COVID-19. We have heard a lot about time-sensitive issues around testing, PPE, ventilators, how and now drug candidates, vaccine development. And today we will focus especially on technology that could be developed or accelerated within the next 3 to 6 months. So the idea is really to look at what we can do now to address the needs of New York and the world post-COVID. So it's very impressive, and I think I'm not alone there, to see how quickly people can move in a time of such great need. Many of my colleagues have noted that across the scientific realm, competition is on the back burner with collaborations thriving locally, nationally, internationally, which is amazing. And it's something that hopefully we can capitalize on, and not just in the biomedical field. So today we will hear from 5 inventors and their technologies that are at different stages of development, but they all have great potential to be repurposed and thereby contribute to solving the current crisis. Examples that will be shared today are 3 different sensing technologies that are being repurposed for coronavirus detection, the development of smart surface coatings that prevent viruses from sticking and surviving, and even rethinking PPE materials in anticipation of the enormous waste crisis that will follow from the huge additional usage of plastic materials by medical workers. So today we would like to learn about other inventions of this type and explore how we can help to develop and co-fund them and to contribute to the needs for post-COVID New York. So thank you everybody for filling out the pre-webinar questionnaire. As a result, we have a pretty good idea of who you are. We're currently represented, I think a lot of people have dialed in. We've got 22 institutions represented and 15 companies. So that's really wonderful. And you also give us an idea of what you already know about us. So some of you work in the ASRC, some of you have been to the ASRC for events, and others don't know anything about it. So we felt that this was an opportunity to briefly explain who we are, what we do, and then also explain what the Sensor CAT is, so the Center for Advanced Technology that can help fund some of these projects. So the ASRC, I have to say, I genuinely miss this building. Not being able to go in every day and work with the people there is tough. For all of us. It's a fantastic state-of-the-art research facility. It's located in Harlem. As you can see on the picture, you're looking down towards Central Park and Midtown. I'm clearly biased, but I really believe there's no better place to do interdisciplinary science anywhere. We're actually currently in the middle of celebrating our 5-year anniversary at the ASRC, and we're doing this through a series of events and workshops. And the next big one was actually planned for May. But we are working hard to move this one online because clearly we can't do this in person. But please watch this space and join us for celebratory events coming up over the next few months. So during these 5 years, we've got the building up and running. We've recruited fantastic people and are hitting good metrics, as you can see on the slide. The building is an experiment in its own right in interdisciplinary working, in that the 20 labs in the building have completely different focus areas and range from fundamental to applied research that ranges from nanoscience, photonics, structural biology to neuroscience and environmental science. And this is a combination that you don't really find anywhere else. So it gives us unique opportunities. We also have 15 high-end core facilities ranging from nanofabrication, live cell imaging, surface analysis, as well as a dedicated next generation sensor lab and others. And importantly, we're open to all at CUNY, but also to external researchers from academia and industry. So these facilities have already, each have collectively already thousands of users and who benefit from our facilities. So then, what is the Sensor CAT? So we were lucky enough, I guess New York State recognized the uniqueness of our opportunity here. And we were awarded a research center. There's 15 of these throughout New York State. They all have different focus areas, and ours is specifically focused on sensor technologies. Tavis is the business director of the Sensor CAT, and he will give you more detail about exactly how it works and how you can get funded from this. But just to briefly give you an idea, at the ASRC, we're exceptionally well placed to innovate in biomedical and environmental sensing, for example, by combining new concepts in deployable environmental sensors, ultra-sensitive sensing, genetically encoded sensors, and new nanoscale sensing modalities. So, Gia, I see I have to stop. So I'm just going to say, in the light of the current COVID crisis, it is clear that sensor technology is critical. We need to know where the virus is, whether it sticks to objects we touch, how to remove them, how to monitor patients and ourselves, and importantly, also how to prevent and protect ourselves in the future. So in conclusion, ASRC and the CAT are ready to work with you. We're committed to exploring what can be done to support and advance COVID research. Gia, over to you.
Thank you, Ryan. So next we'll introduce Dr. Nina Gray, the Executive Director at the ASRC. Nina?
Thank you, Gia, and thank you, Ryan and Tavis, for organizing this. I want to join you, and also thanking everybody else for being here today to talk about this very important and urgent topic. And what I wanted to say to start off with is that it's part of the ASRC's vision and mission to apply interdisciplinary research and science to tackle the problems that are most important to society. And today it's very clear that that problem is COVID-19. And we've already started to do, in our own small way so far, a few things to try to help with these efforts. I think everybody is trying to make some small difference. So while we were in the process of ramping down our normal research operations, our labs and our core facilities set aside gloves, masks, face shields, gowns, whatever they could find that would be considered vital personal protective equipment. And we donated those to the nearby Harlem Hospital, which is part of New York City Health and Hospitals public healthcare system. We also donated our 3D printers to an organization called Hack Manhattan and a group called New York City Makes PPE, in order to help them increase their ability to generate new face shields and other PPE for nursing homes, for hospitals, for EMTs throughout the New York City area. So far, they've been able to deliver nearly 6,000 face shields, for example, across 4 of the 5 boroughs. And if you look at the top picture on the left, that is their first delivery of face shields, which went to Elmhurst Hospital in Queens. And while it's wonderful to be able to contribute in these ways, we really feel like the ASRC has a lot more to offer and can contribute in a much bigger way by leveraging its resources and its facility to spur new research that's related to COVID-19 towards things like detection, mitigation, protection, and vaccination. I also want to point out that part of being socially responsible in pursuing this research is that we balance the risks versus the possible rewards, and that we continue to protect the health of our essential personnel through social distancing while seeking to activate the most promising research on COVID-19. And I think that's why we're all here today, is to try to find that balance and move the research forward. I also want to point out that there are a number of funding opportunities available out there from the NSF, the NIH, DoD, other federal and non-federal agencies. And if any of you out there are interested in applying for those and think that the ASRC might be a good fit to partner with for those applications, that I encourage you to reach out to me to discuss those opportunities before you apply. And I think what we're all here for now, our next speaker will start to address how the Sensor CAT is going to be one of those funding agencies as well for this effort. So thank you all.
Thank you, Nina. So next we have Tavis Excel, the Business Development Director for the ASRC Sensor CAT. Tavis?
Thank you, Gia, and thank you, Nina, for that introduction and your leadership in the actions being taken by the ASRC to support those hero medical workers that are on the front lines of this crisis. So in addition to the PPE that Nina just mentioned, the topic of ventilators is certainly one that we have all been hearing about in the news, and I want to start today by asking for your efforts, in addition to those of the ASRC Sensor CAT, in considering to help us search online and within your personal networks for a critical component that we are now looking for that is critical to ventilator function, and that is pressure sensors. So as you see illustrated in the slide, New York State, together with the US federal government, is in search of a specific kind of sensor manufactured by Honeywell, which is now part of the new ventilator design, which has started to be manufactured rapidly at scale. Honeywell, of course, is as a manufacturer, is trying to make as many of these as possible. But the reason we're coming out to you and why they've asked us to promote this is that there are many of these sensors circulating on the market that have been used for years, and potentially with your help, we could locate stockpiles of those sensors which might be accessed and or repurposed. We will send out further details about this in the follow-up to the webinar. The city and federal government is looking for an estimated 30,000 of these, but of course, they will take what they can get. Now that I mentioned that, I just want to go ahead and jump into specifically what the Sensor CAT is doing in regards to support for and around this call to action. As Ryan mentioned at the beginning of this hour, our goal is to mobilize, facilitate, and quickly increase the impact of efforts you, as the scientific, industrial, and entrepreneurial community are making to think about and tackle problems related to COVID-19. So the Sensor CAT, together with our counterparts at the New York State level, are looking for COVID-related projects that can quickly be put into action with the potential of producing well-defined results in a 3 to 5 month time period. So the ASRC leadership, together with Nina and Ryan and our staff, together with the CAT and New York State, are already taking necessary steps to move towards allowing restricted, safe access to instrumentation and labs in order to facilitate company projects like you'll hear from today. Although short-term projects and results are certainly what we are all hoping for, however, as the CAT, we understand and we want to make clear that funding is open to longer-term projects as well. I think we all understand that the likelihood of this virus or another returning is high, and long-term efforts to mitigate the current crisis and prevent future outbreaks and needless loss of human life are just as important. So now I want to focus a little bit more on the mechanisms that the CAT can employ in respect to focus on these COVID projects. So as you look at the current slide on the screen, the diagram at the bottom, I want to just reiterate the function of our CAT, which is funded by New York State, is to support entrepreneurial development of new sensor-related technology with collaborative partnerships between New York companies and CUNY scientists. So therefore, as you see, let's say for instance, a New York company connects with me as the CAT director with a project that requires use of ASRC facilities. We work together on a budget defined for a scope of work on a specific timeline. Then the CAT decides based on this information, as well as the amount and availability of company funding for investment, how much matching funds should be awarded to the project in support of this effort. If the company is not already partnered with a CUNY researcher with expertise either in this field of focus or use of the instrumentation at ASRC that's required, we pair them together. So CAT matching funds are then awarded to this partnership within CUNY, and their use dictated by the already defined scope of work, as well as overseen by myself as the business director. So quickly, what do typical projects consist of and CAT companies look like? So typically companies are looking to design, build, and test new product prototypes. These companies tend to be small startups to medium-sized companies. However, many researchers at CUNY are also partnered with major large corporations that we can support as well. However, I will say larger matching funds, funding opportunities of, let's say, around 50% matching funds are dedicated to those companies that require the most economic support and demonstrate highly viable projects. So if a company has a funding issue, I also want to make clear that the CAT is also here to play a role in support of finding and acquiring funding. So just like Nina mentioned, there are opportunities out there at the state and federal levels that the CAT will work with you together in support of gaining that funding in order to use. So one question we know that is on every company's mind is also that about company IP protection. This is the most valuable asset to a company, and I want to stress here that you as a company own your IP, and we will take measures to legally protect the company in the process of forming our partnership so this does not become an issue. Again, the idea here with the CAT is to support the growth of business in New York State. So I've talked about how the CAT itself functions and can support New York companies, but I also want to highlight quickly just how the CAT is at the center of a network of organizations working together to support commercialization of new technology. So quickly, you see at the beginning of this slide, CAT is within an institution of organizations, of course, not all of which could be listed here, but organizations working to organically build startups within the CUNY network. Now, as you see coming out from that, we're also involved with New York State funding programs, as well as other institutions in the New York City and regional area. If you have any specific questions further about how this CAT network could be identified and or utilized, I'm happy to answer those in the panel discussion as well. So how do you get in touch with the CAT? What you will find if you don't already have it is our contact information, which will be provided to you following this meeting. So we'd encourage you to go ahead and send us an email with an overview of a project related to COVID-19, including sensor technology that might be viable based on what I've just told you. If you don't have a New York company partner, we will work to match you with one. So don't feel like that is a limiting factor here. After that process, we will review these with our internal CAT review committee and look forward to creating partnerships out of these viable projects. So next, I want to give a brief example of how the CAT and ASRC, again, as Ryan mentioned in the beginning, are uniquely positioned to work on sensor-related technology projects. Gia, if you don't mind going to the next slide. So unfortunately today, I just want to go ahead and say that our colleague, Dr. Ricardo Toledo-Crow, is actually been afflicted with the COVID virus. He tells us that he is safely recovering at home. So I don't want to go into too much detail specifically about his sensor lab, which he is building up on the 5th floor of the Environmental Science Initiative at ASRC. But what I do want to say is basically Ricardo has been building up a lab where we can, together with the CAT projects, work on building low-cost sensors. So he has the means to do fabrication, as you see on this slide, as well as build electronics and also software firmware for those sensor prototypes. I just want to also point out, based on what Nina just said there, you see that Ricardo did have 3D printing capabilities, and those have also been donated in the project, which Nina mentioned. So at this point in time, I want to hand it back to Gia and move on to the next part of this webinar, which is our company pitches. Thank you.
Thank you, Tavis. So now we'll hear from our 5 researchers from startups and industry on what they're doing to answer this urgent call for research against COVID-19. So we're going to start with Dr. Jacob Trevino from Chameleon.
Thank you, Gia, and thank you, Tavis and Ryan and everyone else who was involved in putting this together. It's really important and very helpful to put everyone in the same space and have a conversation. So my name is Jacob Trevino. I'm the principal scientist at a startup company in Brooklyn called Chameleon. We have developed a platform technology that is used to do point of care detection for molecular diagnosis. So we pick a particular target analyte that we're interested in, and then we take this platform technology and adapt it for that. So our technology has two components. One is a highly selective molecular receptor. The other is a nanophotonic colorimetric platform sensor. And so the way we integrate those two is kind of where our IP lies, and it's a very unique way and gives us some advantages over other technologies, mainly speed and ease of use. And we've been using this technology for a couple different applications. So we started as a date rape drug detection company called Drink Savvy. We've recently moved more into the biomedical space, which is where the rebranding of Chameleon came in. And so more recently, we've been working on the detection of cerebral spinal fluid for people coming into the ED with traumatic brain injuries coming from an accident. And then also for neurosurgeons in the middle of surgery, they can find if there's been indeed a leak. They protruded the spinal sac until they're leaking fluid. So it's a way for them to know that there's a problem. You can see an example here of our sensor, sort of the final design. And the colors that you see are actually real colors that our sensors generate. So there's a positive and a negative example here. And the idea is this works for our cerebral spinal fluid. Once the solution is added, the sample, whether it's blood, urine, saliva, whatever, the transition happens in under a minute. So it's very rapid in response. So for COVID-19 specifically, as I said, our platform technology is very easily adapted for other molecules. And so in this case, we're looking at the SARS-CoV-2 antigens, specifically the N protein and the S protein. And so we're in the middle of adapting that technology. And so the idea here is to have a very cheap, very easy to use sensor that everyone can have in their home and everyone can get tested. Regarding how you can help and what we need, we really need access to nanofabrication facilities for the colorimetric part, characterization facilities of how the binding works. We'd be happy to collaborate with people who have expertise in the structure and behavior of the virus itself, and anyone who has experience in molecular receptors. So we, in particular, are currently working with aptamer technologies, but are also interested in peptides and other technologies. And at that, I'll end.
Thank you, Jacob. So next we have Professor Alan Lyons from College of Staten Island.
Oh, thanks very much. Alexander Greer and I have been working together for almost 10 years now, studying the generation of singlet oxygen in the gas phase from solid surfaces. And we've been able to make quite a bit of progress over these years in understanding that mechanism, and have recently come up with a device that can be used to treat various bacterial-based diseases. Based on that technology, we formed a company, Singlet O2 Therapeutics, LLC, which was awarded phase 1, phase 2 NIH SBIR grants. And we're currently in the process of we've demonstrated that the device is very effective at treating bacterial biofilms, such as those that cause periodontitis. And singlet oxygen is a great way to treat this type of endemic disease that affects perhaps 10% of the worldwide population and causes billions of dollars of damage because it doesn't require the use of antibiotics. The oxygen gas is brought into the excited state through the contact with the sensitizer and the presence of light, and very locally will inactivate the bacteria. So we're in the process of working with Harvard Medical School in miniaturizing the device and using animal models to evaluate its effectiveness in a rat model. Because we received this, the company received this SBIR award, we've had in the process of putting a sub-award into CUNY and take advantage of the Sensor CAT to be able to perform the research here at CUNY and use the matching funds from the Sensor CAT to enhance the product development and be able to understand exactly how the device works and how to optimize its performance. But in light of the SARS-CoV-2 virus, we realized that there's an opportunity for a new product that we're working on, a product that could inactivate the virus. And we hope to be able to redirect the funds from the Sensor CAT to be able to start work on this new product that would be capable of inactivating the virus within 30 seconds. And we've started up a project with Sebastian Poget to help us understand the using just the active virus part or the spike protein to be able to quantify how much time it's required and be able to take advantage of the facilities of the ASRC to be able to speed up and implement the development of the device and hopefully apply for additional funding to support this product development. So looking forward to any feedback and any input that we can get from other people on this call. Thank you.
Thank you, Alan. So next we have Krista Avampado from Beyond Plastics.
Hey, Gia, can you hear me?
Yes.
Excellent, great. Thank you so much. Hi, everybody. Thanks so much for having me join today. I'm really excited to be here. I wanted to tell you about a project called Beyond Plastic. So Nina was talking about all of the PPE that has been graciously and wonderfully donated by the ASRC to help our healthcare workers. Of course, we are facing supply shortages. A lot of doctors and nurses and healthcare workers are being asked to reuse the PPE that they already have. Another problem they're facing is that the raw material for the PPE, most PPE is actually made from woven plastic or non-woven plastic, and that is causing an issue. Most of it is fossil fuel derived. So we also know that that is causing, will have a big environmental impact, not to mention the shortages. And so this solution basically takes a look at both of those problems, the shortage of the raw material used to create the PPE, and also the environmental impact of PPE because it's made from fossil fuel-based plastic, and looks at a brand new solution that actually grows on demand a molecule that is biodegradable from a renewable resource and actually can be used with the same machinery to create PPE. So a company that creates non-woven fabric or creates PPE as a whole doesn't have to retool their machinery in order to be able to use this molecule. It is very, very similar to polypropylene, which is the type of plastic that we actually use for our yogurt containers. You find it all over our grocery store. It's the exact same molecule. This molecule would actually replace polypropylene and be biodegradable and come from a renewable resource. So the science is actually coming from a postdoc who we're hoping that will join the ASRC. Right now he's out in Berkeley, and he has been working very hard on this research. And then the idea is to be able to take that scientific research and be able to commercialize it to help create PPE on demand that also doesn't harm the environment. The other issue that of course we face from fossil fuel-based plastics is that fossil fuel and the plastic that's derived from it causes climate change. And there's a lot of research out there showing that as climate change gets worse, it creates a better and better environment for viruses like COVID-19. So the idea with this type of PPE would be to get ahead of it and not compound the problem that would make future pandemics even worse.
Thank you, Krista.
Sure, thank you.
Next, we'll hear from Professor Adam Braunschweig from the ASRC.
Hi, everybody. I'm speaking on behalf of a company that spun out of work from my lab. And what we do is here we develop molecular receptors that bind sugars. So sensors have two elements. They have the sensing element and they have the propagation of the signal. So for example, Jacob was talking about his technology for looking at binding events. Here, we've developed molecules that bind carbohydrates in particular. And carbohydrates are prevalent on the surfaces of viruses, biofilms, etc. And we reasoned that if you could build a molecule that binds the sugars that are prevalent on the surfaces of viruses, you could detect the viruses without needing to rely on antibody tests or RT-PCR assays. Antibody tests are problematic because your body only develops antibodies at late stages of the disease once you've already mounted a response. RT-PCR is problematic because you need a laboratory, it's slow. So we're looking at developing point-of-care diagnostics that don't detect secondary products of the disease of the virus, rather they bind the virus itself. And what these images on your right are showing you is our receptors binding directly to the sugars found on the surfaces of the virus. And that's a very unique space that nobody else is working in. This company started just before the pandemic occurred. And what we're working on right now is develop our proof of concept to show that we can indeed use these molecules as binders or recognition elements. So what we're doing right now is we're leveraging the CUNY ASRC clean room, matching funding from the CAT to build the proof of concept sensors. And what's important here is that this is the recognition element. And once binding occurs, this recognition element can be mounted onto any detection signal propagation platform, whether you want to use a photonic platform, a field effect platform. But what we're doing is directly binding the virus rather than looking at secondary products either mounted as defense by the body, which can lead to false negatives, or nucleotide fragments that may be present in low concentration and require expensive laboratory equipment. So that's our approach, and it's driven by technology licensed out of CUNY and funded in part by the CAT.
Thank you, Adam.
You're welcome.
Next, we'll hear from Dr. James Schultz from Bayer Incorporated. James?
I'm James Schultz, and I'm here representing Bayer Inc. Bayer is an early stage startup that has been developing a low-cost optical infrared camera. Our proprietary thin film coating is a stack of near critical angle spatial filters that have been configured into an integrated photonic device. This device enables sensitive detection of changes in the refractive index of the top layer, which can be structured or functionalized to respond to numerous applications. Our device is primarily composed of an internal light source, a substrate with a thin film coating, and a detector. Our thin film coating stack creates a useful device by first spatially filtering internal light using wave interference and strong refraction so that the light that propagates through the coating is highly spatially structured. Secondly, the coating has a functionalized top layer that acts as a variable mirror, modifying the reflection and transmission ratio proportional to induced changes in the surface refractive index. If there is no externally induced refractive index change on the top layer, the internal light is completely reflected to the detector. But when an externally induced refractive index change takes place on the top layer, the internal light is partially transmitted through the top surface and scattered, with the remaining light being reflected to the detector. We believe that this technology can be repurposed and applied as a clinical antibody-based assay for the direct detection of COVID-19 virus. It has been shown that by mobilizing antibodies of direct detection of antigen boxes, water at the surface of the coating, inducing a surface refractive index change, typically on the order of 10 to the -6. Our technology has a capability to accurately and inexpensively detect this change. This technique for virus detection is well established, but has struggled with commercialization due to the complexity and cost of the optical readout methods. Bayer's technology is a relatively simple nanofabrication process with basic supporting electronics. It can be developed into numerous applications and packaged as an integrated photonic sensor that is low cost and scalable in its manufacturing. We are continuing to explore the development of this device with our partners at the ASRC Nanofabrication Laboratory by recently collaborated with the Sensor CAT to submit a manufacturing grant, which would significantly aid in this effort. We're working to submit an expedited funding request with NSF and DoD. We encourage anyone who's interested in collaboration on this project to contact me. Thank you.
Thank you, James. Okay, so that concludes our pitches from the researchers. And now we can begin our Q&A session featuring our infectious disease specialist, Dr. Norman Sabio, and Ryan and Tavis, whom you've heard from before. But first, we'd love to hear some from Dr. Sabio joining us from Texas today on his outlook of the COVID-19 pandemic. Dr. Sabio?
Good afternoon.
Hello.
Greetings and well wishes to all. It's nice to be here. I hope you are all well, healthy, and safe. It is certainly an honor to be invited and to participate in this webinar. I always feel smarter when I'm around smart people. So thank you. Thank you, Director Ryan, Director Tavis, and Gia. I'm a clinician, but I have worked in research for a brief period of time when I was doing my fellowship at SUNY Stony Brook. And I have done a handful of clinical trials, mostly phase 3 and phase 4, and have not done any compassionate trials like what they're doing now with this hydroxychloroquine and other drugs for COVID-19. COVID-19 is evolving, and we are all learning, not just every day, but probably every time we hear about it or every time we think about it. It's a moving target, and we are at war. So we need to fight, and we need to fight now. And I do appreciate all your efforts to try to kind of be active about this call to action. I'm with you guys, but I will not guess and speculate. I think I will leave that up to the experts with their models and their up-to-date facts. I will talk mostly about what I know as an internist and as infectious disease. I'm a practicing physician for about 35 years, 25 years as internist and infectious disease. I'm also involved in hospital infection control and prevention, as well as community emergency and disaster management. As I perceive this virus, it is an enemy, and truly we're trying to struggle right now to fight it. But we cannot attack what we cannot see. We do not know where it's at. We do not know what it is. We do not know the mechanism of action. We're still learning more about the virus. So I do appreciate all the fidgets about this effort to try to identify them, provide some biodegradable protective gear, and that's pretty innovative. But I can focus more on the prevention and hopefully increase our defense mechanisms. And I don't need to tell you what you already know about trying to enhance your immune system. It's just basically what we need right now to survive this. But we can create barriers. And we already have those barriers: wash, wipe, and avoid. I see this defensive barriers as something that can be layered. From a distance, we avoid people, we wash our hands, and maybe in the area where they penetrate. The penetration of the virus right now, as we know of, is through the airways, through the nose and the mouth, and maybe through the eyes, because the eyes, the tears drains towards the nasal pharynx. So creating a barrier, something that we probably can create just before the penetration of the virus. You've identified how to identify the virus from a distance. I'm more concerned about the patient. I'm not concerned about the results of the vital signs, the monitor results, but I'm concerned about how the patient responds. And we don't have to wait for that. The virus, as we understand, stays about 3 to 4 days in the nasopharynx, and then on the 5th day, on the average, they will have fever. That's when the virus starts to penetrate and wreak havoc and gets into the circulation. So during that time, the first 3 to 4 days, we have an opportunity to hopefully identify how to destroy the virus, whether it's in the nose or in the mouth. Maybe some barrier that we can create. We do have some defense mechanisms, proteases, enzymes in the mouth that destroys bacteria, but it's still not enough. We do have a lot of bacteria in the mouth, and we do have viruses. Now, this coronavirus, we know, has a very thin envelope, as had been described by one of the presenters, and it does not survive alcohol or soap. But I don't think we can continue to have alcohol or soap in our mouth or in our throat or in the nasopharynx for a long period of time unless somebody can design something that's digestible, tolerable, and safe, which can destroy the envelope. Some people have looked at eucalyptus oil, cinnamon oil. I spoke with a dentist a week or so ago, and he mentioned about chlorhexidine and povidone iodine. So I was kind of interested about this periodontitis prevention by one of the presenters. But then again, we are not successful right now in trying to kill the virus successfully with all our efforts right now. So I do appreciate your innovation, your desire to discover something urgently. And I'm with you on that. I will present the clinical aspect. I'm from the receiving end. Obviously, I've been called to action as well. And I've been mobilized. So I probably will not be able to talk about where I am and what I'm actually doing right now. But as a clinician, in my previous life as a reserve officer, I can tell you what my experience was in the clinics, how patients respond, how patients react, and what we actually need. So it's nice to bridge what you scientists do and what you can offer and what I think could be helpful from us, the receiving end, and to give you some feedback of where you need to continue to focus your efforts. So I do appreciate being part of this great team that you've started. Thank you again for having me.
Thank you so much for joining us, Dr. Savio. Okay, so now we will start to take questions from the audience. So the chat box is enabled now, so please type your name, organization, and your question in case we can't address all the questions today, and we will follow up with you offline. So this question, the first one I have was from the registration form. So this is from Dr. Sean Montgomery to Dr. Savio. How could studies using wearable devices like EmotiBit that continuously measure body temperature, PPG, heart rate, respiration, and skin conductance help to better build epidemiological models and treatment decision trees? Dr. Savio?
That's a very interesting concept, but I think that's been done for quite some time now. Astronauts do that. Houston can tell them when they're getting excited, when they're sleepy, when they're having some temperature changes, oxygen levels. Yes, the technology is available. I'm not sure that it's going to be for public use. I think it's for research purposes when we have those monitors to identify how our vaccine volunteers are responding. I think it's a good way to explore that possibility if it can be made affordable and for maybe wider use. It's a very good concept. I'm not too familiar about the technology. That's more for the detection, early diagnosis, and maybe monitoring and following up of cases. So I think it's a good concept, good idea. But right now, when we do not know what this virus can actually do, it probably will mutate and have a much more potent capability or maybe a weaker capability. So right now, monitoring what we have, I think is a good idea. Thank you.
Thank you, Dr. Savio. So the next question is from Misha Simpson from Queen's University. And the question is for Krista. So Krista, aside from PPE, what other potential technology uses are there for your material?
Oh, sure. And actually anything that's made from plastic related to this conversation, if you think about how much plastic we have in healthcare and in hospitals, right? So IV bags, all of the tubing, ventilators, really there's plastic all over our modern healthcare system. And it's actually the whole reason that we can have a modern healthcare system is because of plastic. So plastic absolutely has helped us live happier, healthier, longer lives. The problem is it has such a huge environmental impact. So this molecule actually behaves very similarly to plastic and can be used not only for PPE, but could really be used for lots of other plastic uses, whether they're consumer, and it could be our grocery stores and the packaging of our food, which is now also very important, and the sanitation issues that we're experiencing related to the food supply that we have. And it could be used really for plastic, for tubing, for IV bags, anything that's in a hospital, all of the protective gear that our healthcare workers are using on the front lines. It could be used in any of those reasons. It's just the beads really look almost exactly the same as the polypropylene that's used, and they're used for the same machinery. So it could absolutely definitely has commercial legs, even far beyond the PPE. But of course, the PPE is what we're looking at as the immediate need.
Mm-hmm. Thank you, Chris.
Thanks for the question. Thank you.
So the next question was from the registration form. So this is from Philip Bell to Ryan. Having in mind that the disease should be tackled from an interdisciplinary approach, what is the criteria for making a project or proposal addressing COVID-19 crisis a priority in funding? Ryan?
Yeah, thank you, Belle. Very nice that you could join us. I hadn't actually realized that you would be on the call, so welcome. What we've been doing is set up a number of criteria in terms of urgency, technological feasibility, cost effectiveness, and just fit with the whole crisis. And then another criterion is also that because we're using New York State money, that company involvement is a New York State company. So what we do in terms of fast tracking is short proposals reviewed by two committees, one scientific committee, one feasibility committee, and then we look at whether social distancing can be observed in the labs when these experiments take place, and then we try to fund them and get them going.
Thank you, Ryan. So the next question is from Antonio Meroli. Is there anyone working on surfactants? Acting on surfactant may be the key to save those patients that are dying even if they are assisted with ventilation. When the alveoli collapse, ventilation isn't effective. So who could answer the question about surfactant? Maybe Ryan, do you want to point?
Yeah, I can. I mean, it's a great idea, and I would say that that's the thank you, Dr. Sabio, for also bringing that up, because I think there's a lot of within CUNY actually a lot of knowledge about colloids and surfactants and interfaces. So I think there are a number of people who could get together and think about biodegradable surfactants for this purpose. I don't know of anyone who's specifically looking at it now, but it's certainly a conversation we're going to have. So thank you for that.
Agreed. So one more question from the registration form. This is from Homero Harari. What are the types of partnerships expected? What are the funding options and the timelines? Tavis, can you take this question?
Sure, I'd be happy to, GS. So Homero, thank you for that question. So I think Ryan kind of touched on it there, and maybe I didn't stress it enough, but of course, this being a New York State funded Center for Advanced Technology, they are looking to support New York State companies. That being said, partnerships could look like a lot of different things, and I didn't really have the time necessarily to cover all those different aspects, but whether you are a researcher coming from another university such as Columbia University or internal to CUNY itself, if you have some partnership or relationship with a New York State company and or want us to make that partnership, that would be typically how this would look. We would manage that funding through, through the avenues within the ASRC and CUNY, and the company itself would have access to how that is used. So I think the other part of that question was, what are the funding options and timelines? So that's really varying. I mean, of course, with the COVID-19 crisis, the state is looking for, projects that can be immediately viable. Like I mentioned before, I think as scientists here, we also understand that it takes time in order to address some of these problems and or test out and analyze new technology. So there's a lot of flexibility here, and also when it comes to funding. So, you know, smaller to mid-sized companies, the CAT has the ability to match upwards of 50% of company cash that's invested into a project with the CAT or ASRC. But New York State is giving us flexibility given this crisis, and be happy to chat about it further.
Thank you, Tavis. So the next question is from Kayla Parr from University of Pittsburgh. Question for James. Would your infrared camera technology be able to detect the virus quantitatively or just its presence or absence? If not, any plans to develop a quantitative assay? James?
Yeah, so the repurposed infrared camera technology could potentially be used for quantitative measurements, but we're still looking into it. We're very early stage in this application because our focus has been primarily on developing a low-cost thermal camera up until this point. And that's still focus because thermal cameras have been used in outbreaks in the past to be able to identify people with low-grade fevers, and can be used as early detection of people who are feeling ill. Our technology isn't ready for that application yet, but we are looking into the detection of the virus and hopefully a quantitative assay.
Thank you, James. The next question is from Tamra Schneider. Is there a link or something that the Office of Research could advertise in its weekly lab meeting, which goes out to tens of thousands of CUNY stakeholders? Ryan, do you want to answer that?
Sure. Tamara, thank you very much for that question. I think it relates to what's next. I mean, we're excited about what we're doing today, and we're excited to have, I can see now we have over 100 people joining in here. It suggests there is an appetite. I think the discussion is going well. So we want to make sure that there are ways to keep people informed. We haven't really thought beyond today too much, but we will do that. And I think this is an excellent suggestion. So we will make sure that there are ways to advertise opportunities, because I'm sure I'm not alone in that. I've already learned a bunch of things today that I hadn't thought about before that could be relevant. So, you know, having these conversations is going to be important. So we will be in touch, Tamara. Thank you for that point.
Thank you, Ryan. So the next question was from the registration form as well. And this is from Maya Nair. Are there any companies interested in making anti-fouling materials to study their structural properties? I think Alan could take this question.
So viruses are really small. They're on the order of 40 nanometers. So most of the anti-fouling, so one of the surfaces we're working at is making an active anti-fouling surface that would generate the singlet oxygen that could actively attack and oxidize the virus surface. So that's one approach that we're taking. But nanotextured surfaces are of great interest, but oftentimes the nanotexture is more coarse than the size of the virus. And so extremely tiny surface structures would have to be developed to prevent the virus from adhering. So it's an interesting area for research.
That is very interesting. Thank you, Alan. So next question is from Nina. This question is for Adam Braunschweig. How much of the virus do you think will be needed to be detectable? Does your detector fit with Jacob's reporter system? Oh, she's playing matchmaker here.
Yeah, Jacob and I have had some loose discussions. There's a few things that contribute to sensitivity. There's the inherent association between the receptor and the virus. There's something called multivalency, which we can design into our platform. So, and then there's the sensitivity of the device. So it's a weakest link issue. You can tune the binding of the receptor, and then there's the sensitivity of the device. So whichever is lower of the two is going to be your limit. But what we've seen is that our molecules can detect in the sub-nanomolar concentration range. So it's probably not our molecules that are going to be the limiting factor. What is a factor is selectivity more than sensitivity. So you have a lot of things floating around in blood and in the body. So how do you discriminate one from the other? And we have a solution to that with our molecules. But that's a little too detailed for right now.
Thank you, Adam.
You're welcome.
So one more question. We're actually finishing up. So if you have any questions in the audience, now is the time to type them out. But I have some more from the registration form we'll go through. So what are the current efforts of ASRC to work with stakeholders at the community, state, and federal level? Would this be something Nina could answer?
That's a great question. So we have been working with city officials in terms of donating our PPE. And we are, and I think maybe Tamara's on the line, so she could speak to the larger efforts from CUNY research. But we are also working with other city officials in trying to get this up and running.
Thank you, Nina.
Have you noticed since everyone is eating mostly at home, people are sharing less and less photos of their meals on social?
Okay, and then we have one more question this is from Ryan. So the question is for Dr. Savio. Dr. Savio, do you have any advice on how we can better engage the end user or the medical committee?
Right now, we're getting feedbacks about the outcome of this encounter. The virus is not winning at this time because we have put in a whole lot of efforts. The treatment trials, the vaccination trials, the efforts that we're doing, they call mitigation. The things that we do, we're all involved in this. We're trying to defend ourselves, we're fighting. So it's not just the medical, it's the public. This is a public health nightmare. And they're realizing that there's a whole lot of lessons learned. And we make all this information available. The problem I see is that we sometimes cannot filter out the real from the fake news. The anecdotes, the reports, the trials being done, the stories being propagated, and the more you hear about them, the more they sound like they're probably true. Where in fact, we need to get a good source of information. Right now, all the medical information that we have has to be filtered in. There's a whole lot of trials across the US, across the world, different kinds of reports coming from halfway around the world in Europe. I've been monitoring them, so I've been seeing a whole lot of difference in approach. One thing is unified though, right now, that the virus is still gaining ground, but we're trying to defend ourselves. So what we do and what you guys are doing, I think is very admirable, that you're also helping out, trying to come up with ways to help everybody else. The feedback that you're going to be getting from the medical community is what you see every day. There's transparency. We are going to report everything that we have, but hopefully the report that we get, or the report that we give, will get to the right people and put them together. And this is what they call the model. The assumption that we make is based upon the reports. And I think our government, specifically the task force, is doing an awesome job trying to incorporate all those information, not just from the US, but globally as well. So we will continue to work with you, and we look forward that you can continue to help us. Thank you for the very good question.
Thank you, Dr. Savio. I think that's a perfect timing to end our webinar today. So I'd like to thank everyone for joining us today for the call. And I'd like to thank all of the speakers for making time out of their busy schedule to join us today and prepare for this webinar on such a short notice. From the audience, if you guys have any more questions, please feel free to contact either Ryan, Tavis, or myself. And I'd like to say a big, big thank you to Jasmine Sabio and Camille Santisteban for putting together this webinar on such a short notice and getting everyone together coordinated. Yay. And if you'd like to sign up for our newsletter, please see the link below here. Also, it's in the chat box, so you can copy and paste it. And we hope to see all of you again in our May event coming soon. So this will be a more general launch event about the CAT, and we'll explain more in details about how we can sponsor your fund, your industry partnered academic research. So thank you everyone for a great webinar, and please contact us for any more questions. Thank you.
Thank you.